So at this point, we've read the introduction. We have a general idea of what the paper is about. And here's the next tip or trick for reading scientific papers. What you don't want to do is keep reading the paper in the order in which it was written. So I would not read this paper by picking up at the materials and methods, which is the next section in the paper. Once you get to the introduction, what you want to do is skip ahead, and let me zoom out so you can see the pages a little better. We're going to skip ahead to the very first table or figure in the paper. So I'm going to go down a page, and there it is. And table 1 and figure 1 are presented together. See how they're one next to each other with no words in between. And what I would do is before I read any more in the paper, I would look at the table, I would look at the figure, and I would look at their captions, and I would read that and look at that first. So let's scroll down and look at the caption for figure 1 and see what it says. So usually when you look at a figure caption, the very first sentence, and let's highlight it, tells you what they're saying that figure tells you. Copper alloy equipment reduced concentrations of bacteria on all equipment types. So they're saying that the copper gym equipment had less bacteria on it than the control types. And then everything after that is going to tell you a little bit about what they did and what they're showing you in the figure. So if we highlight that, and let's just highlight it in a different color really quickly. So here they're presenting for you the average bacterial concentration. That's what mean means, the average bacterial concentrations in the bar graph. So that's an average bacterial concentration, that's an average bacterial concentration, that's an average bacterial concentration. And then within each bar is the median, which is the middle value out of all of their samples. So they sampled lots and lots of different 15-pound dumbbells. The average was this bar right here, and the median amount of bacteria is where the triangle is on that bar. So it's just two ways of representing that data. For now, I want you to ignore these p-values, p less than 0.001 for all pairs. We'll talk more about that next week when we talk about data representations. And so if I look at this figure without reading any more of the paper, what you should do is try and decide what you think it shows. So when I look at this, what they're showing me is the average amount of bacteria. Copper equipment is in blue, and then the standard non-copper equipment is in gray. And what I can see very clearly is that by this graph, there's much lower numbers, or there are lower numbers of bacteria on the copper equipment than there is for the standard equipment. So I'm agreeing with what the caption is saying, that the copper equipment has less bacteria. Then I'm going to come up and look at the table, and the table is actually giving me the numbers. So here are the copper components, here are the control components, the non-copper components, and then here are the numbers for the average, the mean, the median, and various types of equipment. So I can compare this 416 for copper attachments to the 6,514 average number of bacteria for the non-copper equipment. And I can again see that's a much lower number for the copper. And then I keep looking at my table. Again, I want you to ignore the p-value and the n for now. We'll talk about that next week. But it also tells me the percent reduction, meaning that going from 416 from 6,514 meant that there was a 94% reduction in the number of bacteria. So again, I look at my figure, I look at my table, I decide what I think it's telling me. It's telling me that the copper equipment has lower bacteria, and now I'm ready to get back in reading. Now I'm not going to go to the materials and methods, which is the next section. I'm going to actually go to the results. So I'm going to get back out of this, go back up a page. So here's materials and methods. Skip past all the materials and methods until I get to the results. And then the results always have subsections. So here's the very first subsection of the results, and let's highlight that. And it says bacterial burden was significantly decreased on high-touch copper alloy surfaces compared with controls. Notice that that matches the caption in figure 1. So this is the section of the results that deals with figure 1. All of the results are paired with a figure or a table. And we can see that even more if we scroll down, because if I look through all of this section of the results, you're going to notice that it references table 1, Table 1, Figure 1. So what they're doing in this section is they're describing the results that are presented in Figure 1. But it makes a lot more sense when you're reading this section if you've already seen the figure and you have an idea of what it shows, and then you can double check that what they're saying matches with what you think. And if it doesn't, go back and look at the figure, compare, and decide if you misunderstood something or if you think they're overstating their claims in their results section. And if I move on, this is the end of this page. I'm going to skip down to the next page. There's the figure itself. And then here's the rest of that section of the results where they're finishing up talking about Figure 1 and Table 1. Now if I look ahead, I've hit the next subsection. So I would stop reading the results at this point because I am at the subsection that details figure number 2, and I haven't looked at figure number 2. I am going to point out that this is titled Characterization of the Most Common Isolates. So they're going to be talking next about characterization of the types of bacteria that they found. But instead, what I would do, so I've looked at the results related to figure 1 and table 1. Now I'm going to go back and look at the materials and methods. So the materials and methods are also typically written in the order of the figures that utilize those particular materials and methods. So what I would do is I would read through until I hit something dealing with characterization of bacteria, because remember that was related to figure 2. So if I look through the materials and methods, right here, this subsection is titled Bacterial Characterization. So I know that is where the materials and methods start to deal with figure 2, and so I'm going to read everything in the materials and methods up until that point. Now let's talk a little bit about the materials and methods and how you would read those. So the materials and methods, there's 2 ways to think about reading them. One way is to understand the experiment itself, and that's how you're typically going to be reading this. Most of the time you can skim through the materials and methods. You might need to know a particular concentration or a particular strain of bacteria they were looking at, but you don't need to know the nitty-gritty details. The other way you read this is if you actually want to repeat what they did. So if you're a researcher yourself and you want to see if what they did is reproducible, then you actually do want to sit down and write down specific concentrations, specific buffers that they used if they're doing something in the lab, particular numbers of people if they were doing human subjects research, those sorts of nitty-gritty details you would really only focus on if you wanted to repeat the paper. So most of the time you can save yourself some time by just skimming the materials and methods enough to understand the underlying experiment, but you don't need to know every single specific thing that they list in there. So if I go back to my materials and methods, again, I would read, and let's keep this highlighted in yellow because it matches figure 1. So here they're talking about where they did it, where they collected samples, the study site, and cleaning regimen. And then here they're talking about how they collected samples and what types of samples they collected from. So the different barbells that they used. So we'll highlight that in yellow. That all deals with figure 1. And then here, if we keep reading that section, they talk about how the samples they took from the different gym equipment was then plated onto bacterial media, petri dishes, and that's how they actually counted the number of bacteria. So the numbers that they got that we saw in that bar graph in figure 1. So I would look at figure 1, determine what I think it says, read the results related to figure 1, make sure that matches what I think, and then skim the materials and methods related to that. But again, now I've hit the next subsection. This is related to figure 2, so I'd stop, and now I'm ready to go and look at figure 2. So I'm going to zoom out, scroll down, there's figure 1, here's figure 2. So I'm going to zoom in here, here's figure 2. I'm going to look at the caption, and it's going to tell me what they found, or sometimes it just tells you what they did, and that's what this case is. So this is characterization of the most common colony morphologies present on copper alloy and control surfaces. So what they're doing here is they're characterizing not just the amount of bacteria that they found, but the types of bacteria that they found on the copper versus the control. So figure 1 was looking at total numbers. Figure 2 is looking at the types of bacteria. And again, I would look at this and I would draw my own conclusions from it. So So I can see that in both cases, the biggest chunk of bacteria, 61% and 75%, were Staphylococcus. And then I can see that there's a slight decrease between the copper and the control. And then that's sort of made up for because there's an increase in the Micrococcus species of bacteria. And then everything else is relatively small, but notice here this Macrococcus is present in the control but not at all in the copper. And then we have a higher amount of Bacillus in the copper and the control. So those are sort of minor changes in percentages. But again, most of it in both cases is still the Staphylococcus. All right, so now I've looked at the figure. I have an idea in my head of what it shows. And so now I'm going to scroll up. So here is where the results for figure 1 left off. And now I'm going to look at the next subsection. And this is the one that relates to figure 2. And so let's highlight that orange, because that's the color we're using for everything related to figure 2. And so then again, I would read through this and I would see if what they say about figure 2 matches what I think, having already looked at the figure and drawn my own conclusions from it. And then to make sure I was understanding it, or if any of this was unclear, I would come back to the methods and I would read over the methods related to that figure. And again, I'm just going to highlight in orange that section. Now you might notice that I've left this last section of the materials and methods unhighlighted, and that's because it's labeled statistical analysis. So when you read a paper, typically if they run statistics on anything, they will put all of the information related to their statistical analysis in one section at the end of the materials and methods. And so this relates to both figures 1 and figure 2. So we'll highlight sort of half of it in the yellow for figure 1 and half of it in the orange for figure 2. So if we kind of zoom out and look at what we've highlighted so far, so here's our introduction in green, and then the materials and methods we've divided into different chunks. There's the yellow chunk here associated with figure 1, and then there's this orange highlighted section associated with figure 2, and then I have the same thing for the results. I have a yellow section that is associated with table 1 and figure 1, and then an orange section that is associated with figure 2. We're going to call this method figure by figure. And that's how I want you to think of reading the biggest chunk of the paper, which is going to be the results and the materials and methods. Instead of reading it straight through, you want to read it starting with the first figure and then the relevant materials and methods and results sections. Move on to figure 2, relevant results and materials and methods, because all of the information that you need to understand that figure is scattered throughout the paper, and you have to sort of piece it back together by looking through and finding what matches up with what. Again, with the results section, this is typically pretty simple because they're going to mention the particular figures in that section, right? So this paragraph specifically mentions figure 2, so I automatically know that this is where they're describing what they did in figure 2. But when you do this, you're focusing on the major results and not trying to understand every single sentence, and that really should be your goal when you read a paper. So that's one tip for reducing the amount of time that it takes you to read a paper while still getting all of the information that you need to. Again, we'll talk more next week about how to actually critique and understand if the way they've presented their data in their figures or the way they've described it in their results actually matches what they're claiming it does. All right, so we'll stop here and we'll just pick back up with the discussion and references in the last video.